For those of you who may not know me, my name is Gabriel Capella, and I am now serving as the director of IDIBELL, and I have the pleasure of hosting today our first IDIBELL Connect webinar that is going to be delivered by Cristina Muñoz-Pinedo. Cristina is faculty in IDIBELL. Her main interest is cell death and metabolism, and accordingly, she is the leader of this corresponding group at IDIBELL. She has had a long interest in this field, mainly focused in cancer. Taking into account the title of the seminar, Inflammation and Other Cell Responses to Microenvironmental Nutrient Shortage, she's going to share her most recent developments into this field. I guess that you are going to talk a lot about immune cells. I leave the floor to Cristina. She's going to share the screen, and let's see how the experiment works.
Thanks, Gabi, for the introduction. And thank you, the IDIBELL seminars team, particularly Lydia, for setting this up, and all of you for being here. Good afternoon or good morning if you're in the States, or good evening if you're somewhere else. I would like to talk to you today about the work that has been part of the PhD thesis of Franziska Püschel, which is work that we've been doing in the past few years in the lab, in which we try to understand how cells react to starvation. Since the background of the people in the audience could be very diverse, I will go very slowly over some concepts. I hope I don't bore you that much. These are the authors of the article that is already published, since this is going to be recorded. I'm only going to talk about published stuff which is included in the article. So in the lab, we want to understand how cells react to not having nutrients, particularly to acute starvation, but also to starvation in general. Why? I hope to convince you that starvation is important, because it occurs in numerous pathologies. And at IDIBELL, we are focused on pathology and how to treat patients, and starvation is involved in several pathologies. For instance, stroke, for instance, heart attack. Also, nutrient sensing is important for multiple pathologies such as diabetes. Now, in cancer, which is our main area of study, starvation occurs mainly in two different contexts. On one hand, when the tumor overgrows the blood vessels, there is a core of the tumor that is starved, and the degree of starvation depends on how close the cells are to the blood vessel. But on the other hand, many of us who work on cancer metabolism have been tried to promote starvation in the tumor cells, to mimic starvation by depriving the tumor cells of essential nutrients. And so anti-metabolic therapies want to promote starvation in the tumor. So we want to understand how this works in order to kill them better with these therapies. Keep in mind that anti-metabolic therapies are very old. In fact, they're part of possibly the first chemotherapy, one of the first chemotherapies that worked and they're still in use, like for instance, fluorouracil. And what these drugs do is to promote starvation of the cancer cell. And in most contexts, this induces cell death. Now, why does this happen? Why do tumor cells rewire their metabolism? And why do we think that cancer starvation or treating them with anti-metabolic drugs would be interesting? Whereas tumors, all of them, or basically all of them that have been studied, rewire their metabolism. Molecularly, this happens because of the transformation itself, meaning that oncogenes and tumor suppressors rewire the metabolism. Like Gerard Evans says, tumor doesn't invent anything. It adapts or hijacks pathways that are already existing in cells. And when a cell is stimulated to proliferate, for instance, it encounters a growth factor, the series of pathways that get activated in order for cells to proliferate or to grow in size, they rewire metabolism in a manner that is more easy for cells to grow by adapting their metabolism. While in the metabolism in a non-proliferating cell is focused on producing ATP or producing the metabolite that cells do on their general day life. If the cells are stimulated to proliferate, metabolism changes. And it changes in the way that they take many more nutrients. They take a lot of glucose. They take other nucleotides if they can. Otherwise, they make the nucleotides from glucose and from other amino acids. They take amino acids. They take as much as they can, but depending on the cell type, many of the internal metabolites, proteins and fatty acids, they make them from scratch, from glucose and from a few amino acids. So when the cell is stimulated to grow, and I have here in green some oncogenic pathways, the fatty acid synthesis is engaged, is accelerated, and this is mediated by mTOR. Glycolysis is particularly enhanced, and this was described by Otto Warburg a long time ago. The mitochondrial pathways are reduced but not inactive, like oxidative phosphorylation respiration is frequently reduced in tumors, which use the glucose for other things like making amino acids through the pentose phosphate pathway, making serine and glycine to make to make nucleotides. I meant nucleotides through the pentose phosphate pathway. And glucose is also used to make proteins because some non-essential amino acids are made from glucose. So these oncogenes promote the metabolic rewiring. mTOR in particular also drives higher protein synthesis for the cells to proliferate, for the cell to grow. Ribosome biogenesis, which is important, and another group at IDIBELL is also studying this pathway, the group of George Thomas and Antonio Gentilella. And of course, every oncogene wants to stimulate nucleotide synthesis. Other well-known oncogenes, such as MYC, they do exactly the same, with some differences depending on which particular protein they stimulate. But the oncogene MYC rewires the cell metabolism to stimulate proliferation, and it does so by stimulating the same pathways. It stimulates glycolysis, particularly anaerobic glycolysis. It stimulates nucleotide synthesis. It stimulates ribosome synthesis and the use of glutamine as well, and fatty acid synthesis. While on the other hand, like in red you see here P10, which is a tumor suppressor, they block this metabolic rewiring in the sense that they work in a completely opposite manner. And for instance, P53 inhibits glycolysis while it stimulates oxidative phosphorylation. What I want to say here is that the reprogramming of metabolism that you usually see in tumors is an intrinsic part of the tumor. It's tied to its proliferation. So the same way that we want to inhibit tumor ability to divide by blocking, for instance, CDKs, we want to inhibit their proliferative metabolism and not cause toxicity, of course. But cells, and particularly tumor cells, frequently encounter that they don't have enough nutrients to proliferate. And all cells in our organism sense nutrients. In animals, the majority of the growth control and proliferation control is driven by growth factors, by hormones. But every cell still senses nutrients. So we have a series of pathways that sense nutrients and that are extremely conserved since even yeast. And for instance, low glucose is sensed by the AMPK, the AMP kinase. Low glucose is also sensed by when there are problems with glycosylation, endoplasmic reticulum stress occurs, and this activates a kinase and the transcription factor ATF4. On the other hand, when amino acids are low, which could happen because there's not enough amino acids in the tumor microenvironment, or because there's not enough glucose and this leads to a secondary loss of amino acids, then mTOR is inactivated. So the cell tries to stop proliferation, stop protein synthesis and DNA synthesis, while at the same time engaging in other pathways to allow its survival to this harsh environment. And for instance, it engages in autophagy, the UPR, the unfolded protein response pathway to promote antioxidant mechanisms. It uses different nutrients like fatty acids if they can. So the aim of these nutrient sensing pathways, the AMPK, mTOR, even P53, and the transcription factor ATF4 and all the unfolded protein response, is to adapt. It's not to die. But if things go wrong and cells are super stimulated to grow because they have oncogenic activation or they lack the tumor suppressors, they then undergo apoptosis or necrosis. They undergo cell death. This is another subject of study in the lab, but I'm not going to talk about it today. So, well, we have been working for years on trying to understand how cells die when they don't have enough nutrients, because that's what we want to do with the tumor cells, like make them believe that they don't have nutrients or actually block their nutrient utilization pathways. Well, this was happening very soon after I joined IDIBELL, one of these internal seminars that usually are presential. I saw Oriol Casanovas presenting the work of Marta Páez from her group, who work here at IDIBELL, are one of the numerous excellent groups that work at IDIBELL. Oriol presented this work in which they showed that anti-angiogenic therapy, which is being used to treat some patients, and there was great promise about it. It deprives, anti-angiogenic therapy, we know, deprives the tumors of nutrients and of oxygen. What they found was that after treating with anti-angiogenic drugs, the tumors shrink, as they should, but soon afterwards, a program would emerge in which cells would revascularize, and this would actually lead to increased invasiveness and, more worrying, enhanced metastasis. So we thought, this is not good. Why is this happening? Is this happening because the tumor that is shrinking senses something, secretes something? This would be, for instance, VEGF, which is a pro-angiogenic factor, and promote revascularization of the tumor. Maybe other proteins, other molecules that the tumor is secreting under these conditions promote enhanced metastasis. So we wonder whether the drugs that we were planning to use, that we want to use, like 2-deoxyglucose, for instance, that inhibits glycolysis, is it possible that they also cause pro-tumorigenic cell-cell effects? These pro-tumorigenic cell-cell effects have been shown with plenty of chemotherapeutic agents. It wouldn't be that strange that anti-metabolic drugs produce the same effect. At the same time, this could mean that hypoglycemia already present in a tumor that doesn't have enough blood vessels promotes pro-tumorigenic cell-cell effects. So we set up to study this. And what we wanted to know is, what do tumor cells secrete when they don't have enough nutrients, when they have metabolic stress? It was known that metabolic stress caused by hypoxia or by not having enough nutrients promotes the release of VEGF that promotes angiogenesis. This was known. And actually, this was mediated by one of our favorite transcription factors, ATF4, which also mediates cell death in the tumor cells when they don't have enough nutrients. We set up to investigate whether perhaps VEGF was not the only thing that the tumors were secreting. They could be secreting other molecules. Perhaps they would promote more invasion and migration, like in the case of anti-angiogenic drugs, or perhaps they would produce more tumor growth, or perhaps they would sensitize cells to the immune attack, or the other way around, they could keep the immune cells away. Thinking of the environment of the tumor, the immune cells are important, and the stroma is also important. So what we set up to study is what proteins do cells release when they are starved. And this is the 3 parts of my talk, in which we will study first what proteins cells release when they're starved, what are the cell-cell consequences of starvation, and what are the signaling pathways that are triggered upon starvation. So to study this, the first thing that we did was to take 3 different cell lines, A549, HeLa, Rh4 rhabdomyosarcoma cells, and subjected them to glucose deprivation, some of them to 0, some of them to 2 millimolar glucose for different times, so that there was no cell death. We wanted to take the supernatants of these cells before they die, so we exclude any possible effects of the cells dying, in which what we would see in the medium. And then we subjected them to these type of studies, but we analyzed 1,000 proteins, 3 cell lines under different conditions. In some of these cells, what we found was that either treating with 2-deoxyglucose in glucose-rich medium, so inhibiting their glycolysis by treating with 2-deoxyglucose, or reducing the glucose in the medium would change what these cells secreted. And some pathways that changed were related to how cells communicate with the microenvironment. For instance, cytokine receptor interaction, chemokine signaling, and MAP kinase signaling, which are involved in chemokine and cytokine sensing and secretion. Also, some pathways were related to rewiring of the tissue, to tissue remodeling. We decided to focus on one because there's a lot of interesting things, but we focused on one of the pathways that was inflammation, and we focused on the lung cancer cells and trying to understand what these cells secrete when they lack nutrients that are related to, could be related to inflammation. So in this specific arrays, we found a series of proteins that are up when cells that are secreted by cells when they lack glucose, or when they're treated with 2-deoxyglucose, and that are down, some proteins that are down under these conditions. So it's not that glucose deprivation only promotes secretion of proteins, it also downregulates some others. Now, in A549, we studied some of these proteins, not all of them, but we went one by one, and studied the messenger level of some cytokines and chemokines under glucose deprivation conditions or in the normal conditions with time. And we found that by qPCR that some of these molecules have a peak of expression soon after the starvation that then normalizes, while some others have a more delayed expression, like IL-6 in this case. CXCL8 is the gene for interleukin-8, which I'm going to talk about quite a bit. So some proteins were up, different kinetics, but the important thing is whether they are secreted or not. And in order to see whether they were actually secreted from the tumor cells, we collected supernatants for ELISA. And via ELISA immunoanalysis, we observed that A549 cells secrete IL-8, interleukin-8, and IL-6, but not others. We only checked for 4, but some are up, some are not up, and more cytokines that I'm not going to show here today are actually secreted by cells upon glucose deprivation. But because there were many proteins, we focused on 2 of them because they are very important in cancer. These are interleukin-8, gene name CXCL8, which is a chemokine, and IL-6, which is an inflammatory cytokine. They're very different proteins, even though their names are very similar. But CXCL8 is a tiny protein, it's a chemotactic factor. It brings on neutrophils mostly, but it could be chemotactic for other cell types. It's also an angiogenic factor, and this is what was very interesting for us, because angiogenesis is known to be a response to starvation. But also IL-8 is very associated to resistance and poor prognosis of lung cancer and of many other tumors, up to the point that there have been clinical trials, and there will be more probably, using anti-IL-8 antibodies for cancer. The other cytokine that we focus on for this study is IL-6, which shows a different kinetic in mRNA, but same kinetic in release. And that is involved in inflammation. As we know, there are clinical trials and they're going to try to block it for COVID-19. It's involved in a variety of cell-cell responses in cancer, tumor, stroma, crosstalk. It's also very heavily involved in tumor growth and survival. And it's one of the molecules that is secreted to mediate cachexia. Like it's one of the molecules that is known that the tumor secretes that promote a systemic weight loss. So this was very interesting for us because it suggests that starvation in the tumor, in the microenvironment of the tumor, can make the tumor cells secrete molecules that rewire systemic metabolism. And this is what somebody else in our lab is studying now. And I can tell you about it in the next seminar, hopefully. And so we focused on IL-8 and IL-6. IL-8 in particular, we studied it in multiple cell lines, its release. And we can see that the release depends on the amount of glucose present in the medium. The less glucose, the more release. This is normal medium. We have to keep in mind that many tumor cells already secrete a lot of IL-8. This is a part of the oncogenic transformation with RAS, for instance, but many other oncogenes promote interleukin-8 secretion. What we see is that when we deprive the cells of glucose, there's more secretion. This is what happens when we deprive them of glucose. And by the way, the same thing happened with IL-6, pretty much the same kinetics. I'm not going to go over that because it's the same. But besides glucose deprivation, we're interested in what happens to the tumor cells when we treat them with anti-metabolic drugs. Like, is this an effect of not having nutrients, or is this a common response to starvation or drugs that promote starvation? So we saw that if we treated the cells, in this case, it's HeLa or A549 or H460 lung cancer cells. If we treat them with 2-deoxyglucose, we saw that these cells that don't die with 2-deoxyglucose, they still secrete a lot of interleukin-8 upon treatment with 2-deoxyglucose. With metformin, metformin is a completely different drug that is used for diabetes, and that many groups, including what at the Catalan Institute of Oncology, are doing clinical trials for cancer. And this drug is known to inhibit mitochondrial metabolism. And these lung cancer cells are very resistant to treatment with metformin. You need very high levels of metformin on the range of millimolar to actually kill them. But at lower concentrations, we already saw that they were inducing the IL-8 release and interleukin-6 release. Again, this is pretty much the same with interleukin-6 and with interleukin-8, which are very different proteins, and they're probably regulated by different signals. Something else that had already been shown in the literature before was that glutamine deprivation also promoted IL-8 release and also IL-6 release, suggesting that this is not exclusive of glucose metabolism, which we know also by metformin because it regulates mitochondrial metabolism. So glutamine deprivation also promotes IL-8 release. We don't know about other drugs that may are being tested in clinical trials, like drugs that inhibit glutamine metabolism. We haven't tested them. We tested complete starvation with a buffer, HBSS, and in this case, there's no release of the cytokines. So this seems to be a general response, but this is exclusive of cancer. And one would say that there's nothing that is exclusive of cancer, but we want to use the things that are more used by cancer. But we also checked whether primary cells would secrete cytokines. And the answer is yes, but not to the same extent. And we tried primary epithelial cells, which in this case are from lung epithelial, human bronchial epithelial cells. And we see that they secrete MCSF, for instance, when they are subjected to glucose deprivation. Also IL-6, IL-8, CXL1, not to the same extent that we would see in cancer cells, because also their reaction to not having nutrients is also much milder. They don't die. They just cope with the stress while the tumor cells die. With 2-deoxyglucose, however, it was different. Not only they did not secrete cytokines, but the cytokine secretion was reduced. And this is probably part of the reason that we want to use 2-deoxyglucose, because the reaction of the tumor cells and the normal cells is different. But indeed, primary cells also secrete cytokines. And I just saw a seminar from a group in Ireland that they're also observing the same effect, the same phenomenon from primary immune cells. So it could be that this is kind of universal, that cells secrete cytokines when starved. Now, what are the cell-cell consequences of starvation? What happens when the cells are starved and they secrete this pool of cytokines? So in order to investigate this, we prepared conditioned medium from cells that had been either incubated with glucose or incubated without glucose for 24 hours. Now, this conditioned medium, we could use it like that, but the problem is that this conditioned medium would contain glucose or would not contain glucose. And then also, if it didn't contain glucose, then the cells would have exhausted some other nutrients. So we preferred a different approach as well, which is to run the medium through filters of 3 kilodalton, keep whatever is on top of the filter, and now replenish the medium with fresh medium this way, and then wash it again. This way, the medium would contain the proteins, or the fraction larger than 3 kilodalton from the conditioned medium, but the nutrients would be exactly the same in one condition or the other. Also, we did some experiments with the filtrate, thinking that perhaps some effects of the conditioned medium would be because of the metabolites that the cells secrete or stop secreting under these circumstances. This is relevant because in the past few years, it has become more apparent that the tumors also communicate with the stroma and with immune cells through metabolites, either by secreting metabolites like lactate, or by depriving the tumor cells or the stroma or the T cells of metabolites such as tryptophan, as you know that they do through overexpression of ID1, meaning that the communication of the tumor cells with the stroma and with the immune cells could be due to metabolites. So in order to find out, in order to remove the metabolites, we wash the medium. So we're only studying the proteins or the exosomes or big particles that are in the medium. So we took our conditioned mediums from A549 cells and we applied it to other A549 cells. And we didn't see any relevant effects on cell death, that was the thing that we've been studying the most, or migration or invasion through matrigel. We did see some small induction of proliferation, that the medium from starved cells slightly promoted proliferation of other A549 cells, but this was small. Now, we also saw some effects on proliferation of HUVEC, which are endothelial cells. The effects are small, but considering that the medium contains IL-8 and also VEGF and also other pro-angiogenic factors, this was not very surprising. Now, the effect is much more clear when you compare medium without from starved cells with a minus here with medium from happy cells, and you see that you can see that the migration of the HUVECs through a membrane is much higher. It's stimulated by the conditioned medium that comes from starved cells. This was not unexpected because of it's known that medium from starved cells promotes angiogenesis. Now, we also wanted to see whether there were effects on the immune cells. And we didn't see effect on T cells, for our disappointment, because we thought this medium could perhaps be keeping T cells away, but it wasn't in vitro. We studied migration of natural killers and B cells towards the medium from starved cells. And we saw a trend, medium from starved cells attracted a little bit natural killer cells, but it was a very small effect. Now with B cells, it was much more obvious that medium from starved cells would attract B cells, and we don't know why or what for. And in vivo, we haven't seen the same effect with B cells, but in vitro, it's very clear that medium from starved cancer cells induce migration of B cells. Now, the most obvious effect was with macrophage-like. I know that they're not macrophage, and people complain about this a lot, that we call macrophage-like the THP1 cells, but they are cells that, when differentiated, they resemble macrophages. And we differentiated them, and they migrate towards the medium from starved cells, while they don't migrate, or very little, to normal medium from A549 cells, suggesting that macrophages migrate towards tumors that are starved. In order to find a more in vivo-like approach to analyze this, the group of John Landricci and Sandrine Marchetti analyzed conditioned medium from CT26 cells, colon cancer cells that had been treated with 2-deoxyglucose. They washed this medium to remove the 2-deoxyglucose, and they injected it in the peritoneum of wild-type mice. And what they observed was the conditioned medium of cells treated with 2-deoxyglucose was attracting macrophages to the peritoneal cavity. This wasn't observed with monocytes, and indeed we didn't see any effects on monocytes of our conditioned medium in terms of attraction. There were several varieties of macrophages that were being attracted by the colon cancer cells that had been treated with 2-deoxyglucose. Now, another very important immune cell for immunity of cancer are the neutrophils. Neutrophils are the first responders to an injury, and they are frequently associated with cancer in a negative manner, meaning that more neutrophils usually indicate a bad response of the tumor. They somehow impair the immunity against the tumor. So we purified neutrophils from blood, or also differentiated HL60 cells that resemble neutrophils, And we subjected them to migration analysis with conditioned medium from cells that had been starved. And we saw that neutrophils, primary neutrophils, and HL60, they migrated towards conditioned medium of starved cells. And this could be due to IL-8 in the medium. We observed that this also happened in vivo. The medium from CT26 cells, which are along a colon cancer, mouse colon cancer, treated with 2-deoxyglucose, attracted neutrophils to the injection site. So this may also be relevant in vivo in the tumor context, like a starved tumor could be attracting neutrophils. Now, is this mediated by the IL-6 or by the IL-8? And yes, when Franci took medium from starved cells and neutralized it by using blocking antibody against interleukin-8 or blocking antibody anti-interleukin-6, she observed that the migration of the neutrophils towards the starved medium was impaired. The same happened with medium from A549 and from H460. So the IL-8 that the tumors secrete under starved conditions is actually promoting the recruitment of neutrophils. As a summary of the second part then, what we've seen is that glucose deprivation or treating with metabolic drugs could promote cell death, but also if the cells don't die, either because the dose is not enough or because this happens before cells die, cells secrete a number of cytokines that could have detrimental effects for the patient, meaning that they would tumor would secrete more IL-8, more VEGF. This could lead to angiogenesis. Also, it would secrete IL-8 and some chemokines that would attract the bad part of the immune system, the immune system that we don't want to have there, which is the macrophages, the tumor-associated macrophages, and the neutrophils. Now, it doesn't promote invasion and migration, or at least of the A549 cells that are the ones we've tested. But given the other cytokines that we've observed, it's possible that these cytokines could promote tumor growth. And for instance, CTGF is a growth factor for multiple tumors, including lung cancer. So in general, these proteins that are secreted by the tumor are pro-tumorigenic in general. Now, we don't know what's happening in vivo. We would like to study this in more details in animal models, and this is what Fedra Luciano will be doing in the lab and whenever we can go back to the lab. And the third part of the talk, which is more for people interested in the molecular pathways, is to try to understand what signaling pathways are triggered by starvation, because what we want to do is interfere with these pathways. If we can interfere with the cytokines, but this is a very expensive treatment for a patient. Treating them with anti-IL-8 or anti-IL-6 is expensive, while we hope to find small molecules that can promote the same effect of inhibiting the bad effects of starvation while promoting the good effects of starvation. So we need to analyze the signaling pathways. And always in inflammation, the main regulator of inflammation is NF-kappa B. So we analyzed whether NF-kappa B was actually regulating interleukin-6 and interleukin-8 release. And indeed, NF-kappa B regulates the messenger level and the protein level of these cytokines, IL-8 and IL-6, and other cytokines. Actually, most of them, except for one that I remember of the 7 or 8 that we tested, we saw that NF-kappa B was regulating most of them. But NF-kappa B is not the only pathway, and we wanted to focus on starvation pathways that are known to participate in how cells signal starvation. How cells signal starvation, as I showed you in the beginning, depends on two main pathways. One converges in mTOR, and when there's not enough nutrients, mTOR is inactive. This is to prevent the cells to keep growing or to keep producing proteins, to keep wasting resources and things that are not needed in these times. The other pathway is the unfolded protein response that is engaged when there's stress in the endoplasmic reticulum. And I'll show you in a bit more detail in a few minutes. And this is the integrated stress response. So first we analyze whether nutrient deprivation inactivates mTOR. This is very well characterized, no surprises here. Indeed, glucose deprivation inactivates mTOR. But is this responsible for the cytokine release? Like, would rapamycin and rapalogs also promote the same effect of cytokine secretion? Well, in our hands, in our cells, they don't. Like, we treat the cells with rapamycin, and they don't secrete cytokines. And even if we combine the rapamycin with the glucose deprivation, we see less cytokine secretion. With taurine, that is a different kind of mTOR inhibitor, we saw that it by itself, it could promote cytokine release. This is IL-8, but we saw the same effects with IL-6. But when we add taurine to cells that are starved, there's even less secretion. So the mTOR pathway inactivation, which happens during starvation, it's not sufficient to promote cytokine secretion. So we analyze the other signaling pathway, which is related to endoplasmic reticulum stress. When there are misfolded proteins, for instance, misglycosylated proteins, these pathways are engaged. And the pathway of the EIF2 alpha factor inhibition, the consequences of this pathway in general are to restore homeostasis in the cell when there's not enough nutrients. There are misfolded proteins. So a series of chaperones are being synthesized under these conditions, genes that promote repair of misfolded proteins, also antioxidant response. And also, if the damage is too strong, apoptosis, as we've shown before. So ATF4 and CHOP are two transcription factors that are activated upon endoplasmic reticulum stress. And Francis saw that indeed two branches of the endoplasmic reticulum stress response are activated in A549 cells when we deprive them of glucose. One of them is the XPP1 pathway, and the other one is the ATF4 CHOP pathway. We know that ATF4 mediates cell death in many cancer cells that we've been analyzing, thrombocytoma, HeLa. And what we saw is that ATF4 was also involved in the cytokine secretion. So by removing ATF4, silencing ATF4 with siRNA, we saw that the cytokines are secreted less. Meaning that this starvation response, which is mediated by ATF4, the transcription factor ATF4, mediates cytokine secretion. And we also observed that some of the other cytokines were regulated by ATF4, but not all of them. It's not like NF-kappa B, it's selective for a series of cytokines or chemokines. Interesting was that ATF4, this transcription factor, was also activated by 2-deoxyglucose and by metformin. Meaning that other metabolic stress, like blocking the mitochondrial respiration with metformin, was also inducing ATF4. So ATF4 could be beyond many of the stress of many anti-metabolic drugs. Now, ATF4 was known to regulate the IL-6 promoter, but we wanted to know whether the effect on the interleukin-8 was direct. And for that, we did CHIP experiments and also other experiments which are called DPI ELISA, in which we synthesize oligos and stick them to the bottom of a plate, then run the lysates of our cells, the nuclear extracts of our cells, and see how much of the transcription factor is bound to this synthetic promoter. By doing this analysis, we have found that ATF4 does bind directly a synthetic IL-8 promoter, and we have identified the transcription factor binding sites which is mostly this site in the islet promoter. These are details. And well, we use a control to show that this site is not bound by NF-κB, which indeed binds the site that was described for NF-κB. So this means that ATF4, which is a response to starvation, directly promotes alert pathways in the cells. So this is intertwined. This is evolutionary programmed that when a cell senses that it doesn't have enough nutrients, it releases alert molecules that would then engage the immune system and would change the stroma, promote angiogenesis. Another pathway that was related, which is the ID1 pathway, was activated, but we didn't find that it was involved in islate induction. And you can see the details, but I'll skip them because it was all negative. We didn't observe either any crosstalk between the ATF4 pathway and the NF-κB pathway. These were experiments performed by Francesca Favaro, previous ones by Jaime Redondo as well. We saw here that inhibiting NF-κB with the common inhibitor BAY did not change induction of ATF4, and vice versa. When we silenced ATF4, we did not observe changes in IKB degradation, meaning that these pathways are parallel, but they're not converging, even though we thought that there could be some crosstalk, but we haven't seen this in this context. And to finalize, the last piece of data that I'm going to show you is that silencing these transcription factors, ATF4 and NF-κB, with siRNA, this is not CRISPR, with siRNA, it was sufficient to prevent attraction of B cells, to prevent attraction of neutrophils, and attraction of THP1 cells, which are similar to macrophages, meaning that we should block these two pathways, the routes that lead to activation of NF-κB and to activation of ATF4, in order to prevent this unwanted effect in cancer, which is that cells that are in the center of the tumor, that are deprived of nutrients, are secreting these inflammatory cytokines that we all know that inflammation, these inflammatory cytokines promote inflammation. That's what they do. And inflammation in a tumor is always associated to, well, it's associated to tumor development on its own. But this kind of inflammation is also negative for treatment with checkpoint inhibitors, for instance. And it's also negative. IL-6, for instance, is extremely associated with lack of response to chemotherapy and also radiotherapy. So we want to block these cytokines for cancer, and we'll do more experiments in mice as soon as we can. So as a summary, nutrient deprivation can promote cell death, but also in an ATF4 dependent manner, secretes molecules that can lead to angiogenesis, possibly to tumor growth, although we haven't shown this in vivo yet, and also to neutrophil and macrophage attraction. And there are many more cytokines that could be having other roles, cachexia, other possible roles that we are currently studying as well. I would leave you with a question that is, what is the physiological role of this? Because we know that cancer doesn't invent pathways. What's the physiological role of this response to starvation? It resembles a lot the inflammatory phase of wound healing. So, because when there's a wound, there's a disturbance of the blood flow. It's possible that this program is evolutionarily conserved to promote inflammation and also proliferation and closure of the wound, but we don't know. Another possibility is that this is linked to infection, like starvation promotes perhaps some intracellular pump-like molecules, because also an infective agent depletes nutrients. But also it could be a conserved response to to ischemia. We know that there's ischemia when there's a lot of artificial situations like organ transplant, and we have inflammation and immune cell infiltration. But it could be that, but also during ischemic disease, stroke, there is inflammation. So perhaps this is linked to blood vessel malfunction in ischemia, or even to developmental angiogenesis. These are all hypotheses, and we don't know what the physiological role will be. What we want is that we want to block it for cancer treatment. And I will leave you with the lab, the people who did the work. This is the current lab, and the people who did the work that I presented today are shown in blue. And these are all previous people that participated in the lab: Jaime, Stefania, Blanca, and Rafaela Iurlaro. I'm sorry that I don't say their last names, but there's a lot of people. And multiple labs around the world contributed. And also I want to thank some IDIBELL labs that have helped us with cell lines, with inside techniques, and that they're here. We have fortunately, we have a lot of laboratories in IDIBELL that do great science, and we have benefited from that for this work. So with this, if there are any questions.
Thanks, Cristina. Now you may listen a virtual applause. Imagine it. This is exactly what you deserve for a fantastic seminar. I remind you, all attendees, that the way to further proceed with questions and answers is by turning on your microphone. And then if you want to make a question, and Lydia will do her best to give you the floor.
Okay, it will be great to have questions or comments.
Yeah, I will begin, Cristina, and it's kind of a general question, but how universal do you feel are these findings? You have now so far tested a number of cell lines. I am not completely sure about how complete has been the analysis for all the variables that you've tested. Which is your feeling, and what else should you do now in order to strengthen these observations?
We are focusing on epithelial tumors, but we've also analyzed what happens in a sarcoma. And the fact that the cytokine secretion is also occurring in primary epithelial cells suggests to me that this is applicable to pretty much all cell types, also because of the work that David Finlay that has presented that have observed the same effect even in macrophages, that they respond the same way. But also we know that in neurons during stroke, we know that in stroke there's accumulation of TNF, IL-1, IL-8, IL-6, and nobody knows exactly where it comes from or why. But these cytokines are calling the immune system in stroke to tell them, go to the brain, there's something wrong happening in the brain. So we suspect that this is universal for all cell types, and a similar program would occur in basically all organs, perhaps with different cytokines, and the pattern was not the same in HeLa and in A549, for instance. The pattern is not the same, but it seems to be a pretty universal thing.
And your focus in IL-8.
Yes.
If you are to focus in another chemokine, would you have other, let's say, attractive candidates?
Oh yes. Yes.
Share something.
Okay. If I had unlimited postdoc
resources,
students, I would definitely focus on one that is CTGF, because this has been involved in growth of tumors. It's like tumors secrete growth factors. Some of those are not chemokines, cytokines. They are growth factors, and this one in particular because it's a growth factor. I think also particularly interesting from the point of view of tumor treatment is LIF, which we're working on, and we are working on several of them that seem interesting from the therapeutic point of view. But we're also focusing on the signaling pathways, the kinases that we can inhibit in order to prevent this effect, and block perhaps multiple cytokines, not only one.
And
question?
Yeah, I do have another question,
wow.
And then leave the floor to others. And you were showing us one of the first slides, Oriol Casanova's work, as kind of the triggering for thinking of an alternative hypothesis. Do you feel that this alternative hypothesis is proven, is alive? How would you define this with that type of evidence you've generated?
Yes, well, in the case of anti-angiogenics, there are some data since that work, as we say in Spanish, it has rained a lot, and since then there are a lot of studies that show that indeed there is IL-6 and IL-8 secreted when patients are treated with anti-angiogenic drugs. Now, the mechanism has not been analyzed. It could be the macrophages that are doing that. It could be other cell types, but we think it's a tumor itself that is reacting to the lack of nutrients, not only to the lack of oxygen. But of course, VEGF is involved as well. VEGF is one of the molecules that we observed. So we're focusing very much on the glucose response, but with anti-angiogenics, there's not only glucose, there's also oxygen deprivation, and there's also deprivation of every other nutrient. So we think it's pretty much the same phenomenon. It's just we're studying it very much more molecularly.
Okay.
I was wondering, because you show some data about migration of different immune populations, but I wonder if you really think it is migration, because there are many other characteristics of these immune cells, particularly maybe for neutrophil survival, maybe more another effect, or for the macrophage-like cells that you use, that I'm not sure how comparable they are to actually macrophages or resident macrophages, or so I guess with these cytokine changes that you observe, you will expect changes in the profile or the behavior of these immune cells, and not really, or not only the migration.
Absolutely. We just had to stop somewhere, but absolutely. Francie did, however, do some studies with the help of Enrique, who is in your lab, who's making neutrophils. And what she observed was that there was no obvious effects on neutrophil cell death. Cell death is something that we can measure very easily. Now, other phenomena are not so easy. So, for instance, we haven't looked at cell death of macrophages in this context, or especially what we would love to study is whether these pool of cytokines affect macrophage polarization.
Yeah, that's, I think, for macrophages, polarization and the behavior on the resident macrophages versus the ones that come from the
Of course, for that we need to do in vivo experiments, and indeed Federa's experience in this type of experiments, and we hope to be able to analyze that soon by modulating not a single cytokine, but the regulator of multiple cytokines, ATF4.
Yeah,
that's what we're planning to do.
Yeah, I think it's smarter to go upstream in that sense, not just with one cytokine. But
we'll definitely need your help for that. use your help if you don't mind.
Sure. Okay.
And more questions?
I agree with you, Cristina, that therapy on interleukins and cytokines in general not only is expensive, but also quite unspecific for cancer. At least it may be my own bias, but it hasn't proven very successful. Not only cytokines, chemokines, but all treatments targeting microenvironment so far have failed. So I would focus my question on the intracellular tumoral pathways, ATF4 and NF-kappa B. How actionable are they? Are there any efforts in those two pathways? I haven't heard that any lead compounds are being developed, but I mean, it may be my own ignorance. And then would those be, could that be related to particular oncogenic molecular background of each particular cell or cancer tumor type that you are working with? Say, would a mutational profile help in defining targeting some particular nodes in this reactivity to starvation?
Yes, there are two questions. The first one, I'll answer the second first because I remember now. Oncogenic pathways. We've been trying really hard to see whether this was an effect that was exclusive of RAS cells, because the majority of cell lines in culture that everybody uses are RAS mutated, right? And of course, this is super important for lung cancer. But we haven't found any pattern in terms of RAS mutations or LKB1 mutations or P53 mutations. Now, for the rest, we don't know, but the ones that are well characterized to play a role in lung cancer, we haven't seen a pattern. But the pattern of cytokines that are released is different in different cell types, for sure. We have focused on IL-8 and IL-6 because, and a couple more, because they are absolutely conserved in every cell type that we've looked for. But for sure, the pattern is different and possibly the regulation is different. And for instance, CXCL1, which is a chemokine, we see it up in some cells and down in some others. So yes, there could be a difference based on the oncogenic transformation. There could be that some oncogenic transformations make cells more needy for glucose. They require more glucose, so they respond more just because they need glucose more. And RAS is one of them that makes them very, very susceptible. LKB1 deficiency is another one, but there are more. To how amenable are the NF-kappa B and the ATF4 pathways? Well, ATF4 itself is a transcription factor, and that's a pain. Now, the efforts are upstream of ATF4 in the kinases that are required for its activation. And there are two kinases that are PERK and IDE1, and both of them are now being tested in the context of cancer and also other contexts. GCN2, there are some small molecules for, ID1 is another pathway in the UPR, I'm confused. ID1 is another pathway of the unfolded protein response, and this kinase has several compounds. PERK, there are fewer compounds, but they're being tested in the context of inflammation as well. And GCN2, there, as far as I know, there are no clinical trials to test it in the context of cancer, but the papers that indicate that the unfolded protein response is essential for cancer are very new. We kind of all suspected this, but the mouse models have not been done. And now they are. Now we can see that ATF4 is essential for transformation with MYC. ATF4 is important for transformation with RAS, essential in the context of lung cancer for what is seen. So since these papers just came up, this seems like a new thing. Now, NF-kappa B is very old, and it has been tried for decades, since Michael Carin's, for instance, experiments. And it's not easy to neutralize, to inactivate NF-kappa B, possibly because it's activated by multiple different pathways. So it has been tested, and I don't know whether it hasn't worked or is out of fashion, inactivation of NF-kappa B for cancer. I don't know exactly what happened. Now the UPR, I know it's new, the unfolded protein response.
So thanks. There's time for a last question.
First, thank you, Lydia, for setting up this meeting. This is great. Congratulations, Cristina, for your talk and for your beautiful paper. It was very nice. I kind of agree with Ramon in terms of thinking in therapy, focusing on the pathway in the cancer cells. You said that NF-kappa beta is difficult to inhibit, but I mean, reading the bibliography, there are many inhibitors of the pathway. I wonder if at some point maybe you can test some of those in your cell culture and check if the level of secretion is lower. So that will be one question. And the second is, so how sure are you that the effect that you see is specific of glucose deprivation? Because I mean, of course, the level of glucose influence a lot in oxidative stress. So I wonder if this is a kind of global response to stress of the cell. You think that something else could also induce the secretion of these chemokines and cytokines?
Yeah, well, regarding NF-kappa B, I really can't tell you. I don't know enough about the efforts to target it for cancer, but I will study very soon. Thanks to you for both of your questions. Regarding the specificity for glucose, we know that it's not specific. There's only one cytokine that we found was fairly specific for glucose, but IL-8 and IL-6 are not specific for glucose, and they are induced in response to stress. And in fact, they are induced in response to other types of stress, including chemotherapeutic drugs. And for instance, Taxol is a fantastic stimuli to induce IL-8 and IL-6 from cells. And Taxol does so in an ATF4 mediated manner as well. So even though the unfolded protein response and this transcription factor are generally associated to having misfolded proteins, which is one of the consequences of metabolic stress, but any stress that disturbs proteostasis, bortezomib, possibly even DNA damage, but no, DNA damage there's not a lot. But anything that disturbs proteostasis, which includes Taxol, surprisingly, would activate this pathway and promote cytokine.
I see.
Okay. So thanks so much. I think that we have to close now, and this is coming to an end. So thanks so much, Cristina. Thanks to all you attending, and have a nice weekend.
Thank you for being there.